I just wanted to make sure that everyone can hear me. Yes, okay, thank you. We will start in a minute. Hey, good morning, good afternoon, or whenever you're joining us. Please take your seats. We are about to begin our webinar, Adaptive Biosafety, Succeeding in Contract Research. My name is Katerina, and I'm one of the consultants at Azelia Lab, a small but highly effective biosafety consulting company. At Azelia Labs, we're driven by a passion for protecting people and enabling organizations to conduct research safely, responsibly, and compliantly. We believe that by fostering strong safety cultures and simplifying complex processes, we can optimize your ability to unlock a full potential of scientific discovery. This webinar has been made possible in collaboration with MRIGlobal, a non-profit research institute founded in 1943. MRIGlobal brings together science and engineering to address urgent health and national security challenges, accelerating innovations from concept to real-world application. It is wonderful to see so many familiar names. And if this is your first webinar with us, welcome. This session was inspired by LinkedIn discussion on the biosafety challenges faced by contract research organizations. And that conversation sparked a strong interest in sharing practical insights from both working within the CROs and working with them. Today, we brought together three rockstar experts who will discuss the unique biosafety challenges that CRO face, often quite different from those in academic or biopharma settings. A quick note, this webinar is being recorded and we will share the recording later this week. We also applied to APSA for CM points approval. We will let you know when we receive it. Please feel free to submit your questions and comments in the Q&A or chat. We will love to hear from you after presentation are completed. And all right, let's start. I'm pleased to introduce our first presenter, Dr. Matt Anderson. He is a Senior Biosafety and Biosecurity Officer at MRIGlobal, where he leads comprehensive biosafety and select agent programs supporting contract research across high containment laboratory environments. He brings more than 15 years of experience advising academic, clinical, and private sector research organizations on biosafety program development, risk assessment, regulatory compliance, and facility design. Dr. Anderson combines deep technical expertise with practical real-world insight gained from collaborating directly with scientists to safely enable complex biological research. Dr. Anderson, the floor is yours.
Thank you, Katerina. You see my slides okay?
Yes.
Okay. So first I want to thank Azelia Labs for organizing this and putting it together. Um, I'm happy to present this information to everybody. I have a disclosure statement. Just want to state that the statements and opinions expressed in my presentation are my own and do not necessarily represent those of MRIGlobal, their views and policies. So Katarina provided a little bit of introduction for me. I just wanted to provide a little bit more context. So as she mentioned, I have been spent 16 years as a BSO at academic institutions. I only recently stepped into the CRO world. I started in my current position in February of 2024. This webinar isn't really meant to be a review of regulations and biosafety compliance, but an introduction to biosafety and the role of contract research and contract development and manufacturing organizations, which I'm going to refer to as CROs and CDMOs throughout the presentation. I feel like I've had enough time in my current position to provide perspective and insight about the similarities and differences in biosafety implementation between contract research and academic settings. We chose the title Adaptive Biosafety because that's truly what it takes to implement good biosafety in the dynamic world of contract research and manufacturing. But the concepts and tools we're going to discuss today are not just applicable to CROs and CDMOs. They will also be useful in other dynamic settings like dealing with emerging pathogens, outbreaks, pandemics, or dealing with new and advanced therapies. The question we hope to answer by the end of today is that how do we make good biosafety decisions when information, timelines, and incentives keep changing? In fast-moving research environments like CROs, uncertainty isn't a flaw, it's the starting condition. Traditional biosafety models assume stable protocols, complete data, and time to iterate. That assumption breaks down when the data are evolving, timelines are compressed, and multiple stakeholders are pulling in different directions. Adaptive biosafety isn't about cutting corners, it's about exercising professional judgment under uncertainty. We start conservatively because we don't yet know where risk will emerge, but that conservatism must be paired with planned reassessment, escalating controls when needed, and de-escalating them when the data support it. Those are both professional obligations. What we cannot do is wait for perfect information. Good biosafety decisions are made while uncertainty is still present. This is where CRO biosafety really diverges from academia. In a CRO, you're almost always working on someone else's science, but you're doing it in your facility with your people. That means risk is distributed, but accountability is not. Someone else defines the science, you own the consequences, and judgment can't be outsourced. The client defines the scope, the urgency, and often the initial risk framing, but if something goes wrong, it's your workforce, your infrastructure, and your reputation on the line. So biosafety often isn't the formal risk owner in the contractual sense, but we are absolutely accountable for the outcomes of the decisions we support. That's why biosafety decisions in CROs are never purely technical. They're operational, they're contractual, and yes, they're business decisions. This same dynamic shows up in public-private partnerships, emergency response labs, outbreak settings, where scientific urgency and external pressure shape the conditions for safety decisions. This isn't about blame or contracts. It's about where outcomes land. In a CRO, biosafety sits at the boundary between science we didn't design and the consequences we do own. And this is why we can't outsource judgment to clients, which is what I want to talk about next. So this slide is about a mistake that causes more biosafety failures in CROs than almost anything else, treating client-provided risk as ground truth. On the left is how risk often arrives at the CRO. It's conceptual, early, and design-focused. You have an assay concept, a target agent, an intended use, and usually a limited early data set. None of this is wrong, but little of it reflects how risk actually manifests during execution. On the right is where biosafety responsibility actually lives. Process conditions, how the work is really done. Scale and throughput, what changes when this isn't a prototype or if it still is one. Facility constraints, what the space and equipment actually allow. Workforce exposure, real people, real repetition, real fatigue. and failure modes, the thing that only appear when systems are stressed. This is where uncertainty collapses into consequence. The gap between these two views is not a communication problem, it's a phase of work problem. Design phase, risk describes intent. Execution phase, risk determines controls. Client assessments inform decisions, but controls are set where the work is actually executed, which is inside the CRO. This isn't about mistrusting clients, it's about recognizing that risk becomes real when people, equipment, and scale, and time and pressure enter the system. Judgment cannot be outsourced. Responsibility cannot be either. Once you accept this, adaptive risk assessment becomes unavoidable because execution keeps changing. That's why risk assessment in CROs can't be static. Adaptive risk assessment doesn't mean starting over every week. It means starting conservatively, planning risk reassessment, and being willing to adjust when the data justify it. For example, client wants you to test their device for limited detection with 4 to 5 different risk group 2 organisms. The device is close to the final design. It's a cartridge-based system that is not completely sealed. The conservative approach is to place the device in a biosafety cabinet to contain any potential aerosols or leaks. Standard BSL-2 PPE is recommended, and you also recommend respiratory protection. Staff are instructed to monitor for leaks and report any identified. During testing, there is no observation of leaks from the device, and you can rescind the recommendation for respiratory protection, as there is now a low probability of aerosol generation from the device. This process has to be intentional. If reassessment only happens after incidents, then the model isn't adaptive, it's reactive. So what we need is a feedback loop that assumes uncertainty up front and expects reassessment as part of normal work. What I just showed you wasn't a workaround or an exception. It's the operating model. Adaptive biosafety starts by acknowledging uncertainty. When we don't know everything yet, conservative initial controls are appropriate. As work is actually performed, real-world signals emerge, things that the paper assessment can't fully predict. That information feeds planned reassessment, and not after something goes wrong. It's part of how the system is designed. Judgment is the professional obligation to change course, to escalate or de-escalate when the data justify it. The danger isn't uncertainty, it's freezing the assessment and pretending uncertainty doesn't exist. Once you accept that risk is shaped by how the work actually unfolds, relying on fixed categories alone stops being enough. So risk group and biosafety level tell us what the agent is. They do not tell us how risk emerges once the work is automated, scaled, or enclosed. That translation step from category to control is professional judgment. Consider lentiviral vectors, typically treated as risk group 2 and often mapped cleanly to BSL-2. In first scenario, you have small volume, manual transduction, it's open bench work inside of BSC, trained staff, low throughput. Standard BSL-2 controls are likely appropriate. In the second scenario, you have automated high throughput systems, you have pressurized cartridges and tubing, unknown failure points during the runs. Same agent, but aerosol and exposure risk now depends on the process, not the classification. The risk group didn't change, but the workflow did. Category labels can stay the same, but the controls had to change. This is why adaptive biosafety focuses on how the work is done, not just what the agent is. So we rely on committees to structure safety decisions. But committees don't actually make decisions, judgment does. That judgment leads to real containment and operational decisions, but the problem is what happens to professional judgment when committees operate under pressure from timelines, clients, and mission. Decision quality degrades when urgency compresses thoughtful judgment into speed-driven consensus. As timelines tighten, judgment quietly shifts towards precedent, convenience, or getting to yes rather than the risk as it actually exists. Under pressure, committees can mistake agreement for rigor, allowing familiarity and speed to substitute for critical evaluation. Protecting safety requires predefined options to elevate authority, add expertise, pause decisions, or adjust the controls before judgment collapses into approval by default. Rigor comes from judgment. Pressure quietly reshapes that judgment unless we design for it. So this slide is really about timing, not authority. When biosafety engages early, adaptive biosafety actually enables speed by stabilizing decisions before execution begins. Upfront judgment made when uncertainty is highest prevents the kind of mid-project changes that cause rework, delays, and frustration. Most delays I've seen in CROs weren't caused by safety saying no. They were caused by biosafety not being involved before timelines were established or escalation being needed mid-project. Early engagement isn't just about controls, it's about explaining judgment calls clearly and building trust with technical staff, leadership, and clients so decisions hold when pressure increases. That becomes even more critical as risk and containment levels increase. This slide is about where risk actually lives in high containment CRO work. The facility usually doesn't change. The containment systems are fixed, they're validated, and generally sound. What does change is the pressure, the timelines, throughput, staffing strain, and that pressure flows through people first. As pressure accumulates, the limiting factor here isn't the pathogen, it's whether the system protects human judgment, attention, consistency, and willingness to escalate. And when judgment starts to erode, risk doesn't announce itself with failures, it shows up quietly as signals. That's what I want to look at next. So this is what pressure looks like before anything goes wrong. Under sustained operational pressure, judgment erodes quietly. What we see instead of incidents are small signals. People take shortcuts, there's fatigue, you have near misses, they get corrected instead of being surfaced and addressed. Formally, individually, none of those look catastrophic. In isolation, they're easy to explain away. The danger isn't uncertainty, and it isn't even those signals by themselves. The danger is when the system quietly adapts to pressure by accepting them as normal. So let me show you what that looks like in a real CRO where nothing failed, no one is exposed, and adaptive biosafety intervened before an incident forced the issue. So this is anonymized, but very typical. Nothing catastrophic happened, no exposure, no containment failure. A CRO was running multiple client projects through a single BSL3 suite. Facility design is sound, SOPs are compliant, inspections have gone well. What changed wasn't containment, it was the pressure. Shift coverage was stretched, so senior staff began covering gaps. Training was complete, but the cognitive load was high. Small things started showing up, dawning errors caught by peers, deferred maintenance, informal shortcuts, near misses corrected quietly. The inflection point wasn't an inspection or an incident. It was burnout. Adaptive biosafety allowed intervention before harm occurred. They paused new work intake, rescaled timelines with leadership, and treated staffing strain as a biosafety control failure, not an HR issue. Now, the lesson wasn't about containment, it was about protecting judgment before it collapses. The hardest BSL3 decision is often knowing when to slow down. So most biosafety training programs are built to satisfy regulators, not to support judgment under uncertainty. In CROs, that prepares people for audits. It doesn't prepare them for ambiguous data, evolving workflows, or conflicting priorities. In CROs, we rarely have stable, long-tenure teams. People are often new to the organization, new to the platform, or new to containment, and critical safety decisions are made during execution, not in committee rooms. That means training has to focus on recognizing uncertainty, surfacing concerns early, and knowing when stopping or adjusting work is the right call. Job titles don't predict decision quality under pressure, competency does. So this is where CRO training starts to diverge from academic models and why the same adaptive loop looks very different depending on the environment. If staff aren't trained to recognize uncertainty, the feedback loop breaks. This comparison isn't about sectors, it's just about operating conditions. Academic biosafety training is typically optimized for stability. CRO training is optimized for execution under changing conditions. People are onboarded fast, platforms evolve mid-project, and risk decisions are made at the bench, not during setup. Public health labs live permanently in adaptive mode. They're trained to act reasonably with incomplete data and revise controls as evidence evolves. Reassessment is the operating model. All three of these environments operate within the same adaptive biosafety loop. The difference is how fast the ground is moving beneath you. Adaptive biosafety isn't about where you work, it's about whether your training prepares people to make judgment calls when conditions change. Just want to touch on regulatory complexity. This slide isn't really about memorizing regulations. It's about understanding how regulatory complexity changes biosafety judgment outside of academia. In CROs, regulatory triggers are tied to use, destination, or ownership, not just biosafety risk group. You have GLP, GMP, import permits from Fish and Wildlife, CITES, USDA, CDC. Regulations don't replace each other, they stack. The same work can be subject to NIH guidance, CDC oversight, OSHA requirements, FDA expectations, expert controls, and client-specific frameworks all at the same time. What makes this challenging isn't memorizing regulations, it's recognizing when a regulatory trigger appears mid-project and integrating it into biosafety decisions in real time. Outside academia, biosafety judgment has to integrate safety, law, and contract simultaneously, often under changing conditions. So this is culture and practice. One side is judgment, people, thinking, the ability to pause. On the other side are the incentives, speed, deadlines, billability. Notice there's a slight imbalance that's intentional. The pressure is always on. And the only thing keeping judgment in the game here is leadership, not policy, not committees, active leadership stabilizing the system. Culture is what actually wins when these two are in conflict. If staff don't feel safe pausing or escalating, adaptation never happens. Incentives are always present, but judgment is not. You get exactly the behavior your incentives reward, even if that behavior undermines safety. In the next section, I'm going to talk about culture shock of moving from academia to contract research. So a lot of people experience culture shock when they move from academia to CROs and CDMOs, and I know I did. That discomfort usually isn't because they lack experience, it's because the operating conditions are fundamentally different. Ambiguity is structural, commercial pressure is real, and biosafety judgment gets exercised earlier, faster, and closer to execution. This slide isn't about whether one system is better, it's about understanding why the transition feels hard, even for very experienced professionals. If you're coming from academia, knowing the basics is still essential. Those foundations carry over. What often changes is how fast you have to move from guidance to judgment. Risk assessment becomes more dynamic, and timelines and incentives become part of the risk environment, whether we like it or not. One positive surprise for many BSOs is that leadership is often more directly invested in biosafety outcomes because the business consequences are immediate. The challenge isn't abandoning academic rigor, it's learning how to apply it under different constraints. Think of this less as advice and more like orientation. Judgment is your true north, pace, leadership, and incentives can shift around you, but foundations come with you, but they don't determine the directions. No matter where you work, judgment is where what you're navigating by. Everything else shifts around it. In industry, pace isn't a background condition. It's an active force you have to account for. In CROs, leadership isn't distant. Decisions land somewhere and leaders feel the impact. And foundations matter, but they're not where you stand. They're what you bring with you. Everything I've talked about today lives inside the adaptive loop. Adaptive biosafety starts by acknowledging uncertainty, not trying to eliminate it on paper. Uncertainty isn't a failure of preparation. In fast-moving science, it's the starting condition. That's why conservative initial controls are a feature, not over-engineering. They're an honest response to what we don't yet know. As work begins, the work itself reveals risk. Real processes, workflows, and human behavior show us things no paper assessment can. Adaptive biosafety depends on deliberate reassessment, not reacting after incidents, but intentionally asking whether the risk framework still fits reality. Credibility comes from being willing to de-escalate when the data support it, not just escalate. And none of this works unless judgment is protected under pressure, protected by committees that resist rubber stamping, staff who feel safe surfacing concerns, and leaders who prioritize decision quality when speed and safety conflict. Adaptive biosafety isn't looser, it's more more honest about how science actually works. Whether you're in a CRO, public health lab, or an academic program, if your science is moving fast, this loop applies to you. Thank you for your time, and we'll transition to our next speaker.
Thank you. Thank you very much. I wish I heard this presentation 5 years ago. Then I moved from academia to CRO. We will ask all the questions after presentations, and but now I'd like to invite our next presenter, Dr. Ahrens. Dr. Ahrens was born and raised on the beaches of Southern California, where he earned his undergraduate degrees in physiology and neuroscience at UC San Diego. Then went on to earn his Doctor of Veterinary Medicine from Western University College of Veterinary Medicine in Los Angeles. He then earned his PhD in radiochemistry and cancer imaging at the Iril and Manila Graduate School of Biological Sciences, completed a residency in laboratory animal medicine at the University of Southern California, and then did a postdoctoral fellowship at the City of Hope Beckman Research Institute. Brad spent 6 years as a research veterinarian for Caltech and USC in LA during a 10-year term as the veterinary director for Universal Studios. In 2022, Brad left Southern California and joined LabCorp, when he is now the director of animal welfare and attending veterinarian for the LabCorp Greenfield site. Welcome.
Thanks, Katerina. Can you hear me okay?
Yes. Mm-hmm.
Okay, excellent. So welcome, everyone. Thank you for attending today. So today, as the veterinarian in the group, I'm going to be focusing on, obviously, contract animal research and going at everything more from an animal perspective. So CROs, obviously, they support diverse research needs while, of course, needing to navigate complex biosafety and regulatory environments. So today, I'm going to talk about some of the common issues faced in CRO biosafety, specifically with animals, and discuss some of the strategies to mitigate risk. In the interest of time, I'm just going to highlight a couple of the major ones that I see on a daily basis and some of the ones that I've encountered in my career to hopefully help guide those of you on the webinar in your own institutions. I'd also like to disclose that views and opinions discussed in the webinar are my own and do not reflect those of my employer, LabCorp. So any information is simply for educational purposes. So with that, I'll get started. I'd like to start with a visualization here. So close your eyes and think about this. Picture this. It's 6 o'clock in the morning. You're already behind in your schedule. You've been having a tough week. Maybe your boss isn't thrilled with you, and you're just doing a routine procedure. You're already running late, and you forgot your face shield on the other end of the vivarium. You think, oh, I've done this 1,000 times. I'll be fine just this once. And so you go ahead, you do it just like you've done 1,000 times before, and suddenly something happens and you feel a splash on your face. You think, I'm fine. I didn't even get my eye, I don't think. I'll just wipe it. I'll get done with my task. I can't not spare the time to go down to EH&S and report this again. So this is a scenario that it's a terrifying scenario, especially if working with non-human primates, that this is how biosafety failures start. We're all human, and it's just a really good illustration of how these situations arise and why it's so important to have good standards for biosafety in our facilities. So I'm not going to make you guys raise your hands because we're all virtual here. But you can raise your virtual hands or just play along with me here. How many of you have seen a biosafety issue caused by human behavior? I'm sure all of you, I would hope. Yes. Okay. Obviously. But now, on the other hand, for those of you who do work with animals, how many of you have seen a biosafety issue caused by animal behavior? Okay, a large number of you. But now, kind of getting to the point of my talk today, who was at fault in either of these situations? That's been my experience too. I've certainly seen biosafety issues caused by humans, and I've seen them caused by animals. But who was at fault? And so, what I'd like to present to you today is that I think the one at fault for both of those are the humans, not the animals. Animals are predictable. They're predictably unpredictable, but in essence, they are predictable. We know what they're capable of. We expect the worst when working with animals. We know they have sharp teeth. They can pee at any moment. They are the inherent risks of animals. We understand that. So when we have a biosafety issue in animal research caused by human behavior, that's obviously because of a human. But when we see one caused by animal behavior, we should have seen that ahead of time. That's on us because the animals are predictable. We know how they act. We didn't put good enough measures in place to prevent that in the first place. So I'd like to argue that the human is at fault in both of those situations. When I think about biohazardous animal research, I think of the term controlled chaos. Whenever I hear the term controlled chaos, I always think about the movie Spider-Man 2. It came out a while ago, but if any of you have seen it, there's a guy, a crazy scientist in it, Dr. Octopus, who wants to create a fusion reactor, and he creates this small, essentially nuclear fusion reaction. And he has these mechanical arms that he uses, and these arms, every time it starts spiking out, he'll take one of those arms and he'll push back into the center and try to control the chaos as he sees it. And this is kind of how animal research works. And the more variables that you have, the larger that reaction is, and the more arms that you need to be able to contain all of the uncertainty in that process. So when you are dealing with multiple species, when you're dealing with multiple pathogens in multiple species, when you're dealing with unknown experimental agents, which are inherent of CRO work, and then when you're working with multiple sponsors with very different requirements, all of these things make a fairly large reaction, and you're going to need a lot of arms in order to contain those things. And no matter how much you prepare, some of those things are going to come, and you're going to have to respond to them as they arise. So while I could go on for hours, I wanted to focus on just two of the most significant challenges in CRO animal biosafety that underscore the importance of being adaptive. The first of which is the species portfolio. Every species presents its own challenges. You have monkeys or non-human primates, we have dogs, we have pigs, and we have rodents. Of course, there's plenty more species used in research, but I think these four really illustrate the differences in the species and how we, as biosafety professionals, need to prepare and for those different species and for the uncertainty inherent in all of them. So the first one is primates. Primates obviously require a lot of respect. Everybody on here, I sure hope everybody on the call today knows all about herpes B, you know about tuberculosis, you know about these pathogens that we assume no matter what the source of your primate is, no matter how long it's been in captivity, no matter where it came from, you assume that it carries all of these pathogens. So essentially, and many of these, these are biosafety level 3 pathogens, and you are not wearing biosafety level 3 PPE or taking those kind of precautions. So it can be very scary thinking about that we have these risks that are just inherent in these species, and our PPE level is not always at the same as the level of the risk presented by these species. So of course, in these, bites and scratches always matter. Everything's a big deal because complacency and skipping that step, or in that scenario that I painted at the beginning, complacency can be fatal. If you don't take it seriously and you don't instill the values in the staff to take every possible exposure seriously, the consequences can be fatal. Dogs are a great one in that dogs are friendly, especially, most dogs used in research are going to be beagles. They're adorable, they're sweet, they're friendly. I've never heard of anyone being seriously bit by a beagle in a research environment. It's pretty rare. And their zoonotic risk is very low. We all have pets at home. The risk of a natural zoonosis in a lab raised beagle is next to zero. So, however, there are, depending on the types of studies that are in them, they can, and what sort of pathogens are used, that can, of course, present a biologic risk. And the problem with these dogs is they're adorable. They're cute, they're friendly. As soon as you open up an enclosure, they just want to come and they want to jump on you and they want to play with you. And they love that human interaction. So, and it's disarming for those of you who work with dogs, is that when you work with them, you go, oh, this is just like my dog at home. I want to love it. I want to pet it. I'll wear my PPE. That's fine. But we see a lot more PPE slips and laxity when working with these species because they're so cute that you let your guard down, and you can have exposures that way. Next is pigs. I like to say, here's a picture here of a if anybody's ever seen one, it's a greased pig contest where you have to try to catch a greased pig. I like to think of pigs as operational chaos. They are strong. Many times, if you're working with full-size pigs, they're hundreds of pounds. They're far stronger than a human. They're very smart, and they can be messy. So while they're on the ground and they're not on a perch like a primate would be, they can splash. There's a lot of things that they can do that can present exposures. And unlike dogs, pigs can carry some, moderate zoonotic pathogens naturally that could cause illness in the staff. But then especially with whatever biologics you're working with, that presents obviously a much greater issue in that these very strong animals can easily get through a lot of different types of PPE. And lastly, we have our rodents. Rodents, you're dealing with a problem of scale. So we have thousands of animals when you're doing rodent studies, and a small error can mean a big impact. Interestingly enough, with all 4 of these species, there's also worries of reverse zoonosis, right? We're worried that we can give something to them, especially in the case of primates. Primates can catch human herpes. Primates can catch measles. They can catch a number of TB from humans and then give it back to other humans. They can catch a variety of pathogens from us. Not so much of a concern with dogs or pigs, but with rodents, very much so, especially when you work with immunocompromised animals, that you're almost more worried about them catching something from you than you are from them. However, as with high containment biologic research, it's typically done in rodent models. So when you're working with rodents, they do have sharp teeth, and they're very small. If you're working with needles or sharps, there's always the potential for a needle stick or a cut when working with these animals. And these are typically the ones that are going to have the more dangerous pathogens and the higher containment pathogens. So the second challenge I wanted to present, and it's very similar to what the previous presenter talked about, but it's the unknown. In a CRO, study directors are performing somebody else's research. While we may have the operational expertise, we at the CRO often do not have the scientific expertise or the technical understanding that the creator of the test agent has. We don't necessarily understand the agent, how it was created. So there's a big gap in knowledge there that can create safety issues. On the other hand, at the CRO, typically we have a better operational, usually a better veterinary, and many times a better technical knowledge and experience than the client does. And so when the information lies in 2 different courts, there's gaps on both sides. And these gaps in understanding are what I like to call the unknown. And it's the greatest areas of risk where a client can have unrealistic technical expectations and the CRO can have incomplete understandings of the dangers and the characteristics of the test article. And with that, it can be either intentional or unintentional. So many times in a CRO setting that we are purposefully not told exactly what we're doing or exactly what the mechanism is. And this is a risk that sometimes we accept, but generally we'll try to find middle ground where we can know as much as possible without knowing too much or endangering the potential of the drug or the company. And then unintentional, of course, is that these are experimental drugs. There's inherent unknowns with them and what effects they could have. Of course, we always have human error on either side. This can be a formulation error on the sponsor or the client side, where maybe something's made far more concentrated than it should be, or it can be on the CRO side with dosing, with a misunderstanding of a technical sheet, misunderstanding of a dose formulation instructions. Human error happens, and it can happen on both sides, and it can create a large biosafety issue. So biologic uncertainty in different species is another thing we have to worry about, and that many times when a CRO receives a test article, a lot of times it's never even been in that species. So we're unsure how the species will react. Some may cause neurologic signs and could turn an otherwise perfectly harmless dog aggressive. And it can be shed in different ways, maybe differently than what it was done, PK studies done before that showed maybe different shedding in different species. It can have completely different activity in another species. So there's a lot of biologic uncertainty there. And my advice for this is to trust but verify, right? Put measures in place that will protect the staff and the institution, but you can hopefully trust that they know what they are doing. And at the end of the day, overconfidence is dangerous. You always want to not assume the worst, but assume that there could be something wrong, whether intentional or unintentional, and plan for that. So what do we do about it? So getting to my conclusion here, first and foremost, don't overcomplicate things. With both challenges, it's important to understand and acknowledge what you know and what you don't know. With challenge number one of the species, obviously you want to evaluate risks with consideration for the species. You want to evaluate what are the risks of biting? What are the risks of shedding and having that? It's very difficult to get mouse urine in your eye. It's very easy to get primate urine in your eye, or pig, even with great PPE. So you need to evaluate the risk and consideration of the species and take that in consideration with the test article you're testing. And then challenge number two is the unknown. As I mentioned, what I usually like to say is prepare for the worst, or at least something near it, based on the potential for uncertainty. And some good tips are create simple SOPs. Don't overcomplicate things. Create simple SOPs that are simple enough to follow. I've found that when SOPs are too complicated, people can forget steps. So keep it simple. Train for reality. There's always, when you do your training, some people will train for these perfect situations and exactly what is supposed to happen. But we all know that humans and animals are unpredictable. So you want to train for the reality of that situation and cover your butt. And last, design for human behavior. People will always find a way around engineering control. People will always find ways around PPE. Many people sometimes will just forget or intentionally not wear it. So you need to design for human behavior and make sure you have that in consideration. And lastly, collaborate. You've got experts. We've got a whole panel of experts on this call today, and you all probably have these same experts at each of your institutions as well. The vets are going to have the vets or veterinary staff are going to have great information on pathophysiology of different things, how animals might react, what they've seen before. Your biosafety team, of course, is going to have the most knowledge about how to protect yourself, but then your operations teams are going to know how things actually run. And if there's gaps in any of these processes, we can always fill them, but you want to do that before your study. So the final takeaways that I have is that when you have different species, you have different risks. Don't treat all species the same. When you're dealing with the unknown, you want to trust but verify, and it's essential to be adaptive in your biosafety program, which is conveniently the title of this talk. So in closing, thank you all for your attention. I hope the challenges and solutions I discussed are relevant to your facilities, can help you and your staff be better prepared and be more adaptive in your biosafety programs. And with that, stay safe, respect the animals, and wear your PPE. Thank you.
Excellent. Thank you so much. I see one raised hand, and if it's possible, please submit your questions through chat or Q&A. We now have our final speaker, Jason Clemens, and he's the biocontainment coordinator and responsible official at the University of Florida. But previously, he served as a biosafety officer at National Resilience Incorporated, where he supported biosafety and biocontainment operations at a Department of Defense funded 300,000 square foot biological development and manufacturing facility capable of large-scale BSL-3 containment and located in Alachua, Florida. The site produced live vaccines and oncolytic viral therapies with a drug substance manufacturing capacity up to 2,000-liter scale. Jason's experience bridges academic research environments and large-scale GMP-regulated industrial biomanufacturing. Please join me in welcoming Jason Clemens.
Thanks, Katerina. So let me share here. All right, you should be able to see. So I'm going to talk about the CDMO startup context of adaptive biosafety, which is my background. And so to go into my background a little bit more, I have a background in quality assurance, quality control. I was briefly a state regulator for sterile compounding in Maryland. But for the past 8 years, I've been in biosafety, and I spent 6 of those at the CDMO that Katerina mentioned, National Resilience, which prior to that was Ology Bioservices, and prior to that was Nanotherapeutics. So that alone should tell you, kind of give you a preview of what I'm going to talk about, which is that there's a lot of churn, a lot of change, both in the CRO, CDM space in general, but especially among startups. So I'm going to talk a little bit more about that startup environment and what that looks like and some of the challenges and pressures that come with that. And you'll hear a lot of echoes in what I talk about to what Matt went through as well. I'll talk through some of the adaptations that I think are beneficial, especially in the context of a startup. And this is probably applicable not just to the biotech industry, but outside it as well. And then if there's time, which I suspect there won't be because I want to make sure we have time for questions at the end, I'll go through a case study. So just to give you an overview, very simplified overview of what a contract development and manufacturing organization does. They take some product from a client, and there are both small and large molecule CDMOs and ones that do both. And they take that product and they develop it further so that it can go into clinic, anywhere from phase 1 to commercial development and manufacturing for a particular medicinal product. So you have a tech transfer process that occurs between the client and the company, a lot of process optimization and analytical development. From there, it gets scaled up into manufacturing, where you have an upstream production portion, a downstream processing and purification portion, and analytical testing. So you have everything from large scale work, again, mentioned 2,000-liter bioreactor is the maximum scale the facility I worked at, to lab scale QC testing. And then finally, you take that bulk drug substance and you fill it into a final container as your product that goes to the client and the clinic. And there are biorisk management points throughout this product life cycle. A big part of it, and Matt talked about this a little bit, what the kind of knowledge that you get from your client. And I'll talk a little bit more about that in a bit. Also the culture. The culture can really change between the process development folks and the manufacturing folks quite a bit. There's definitely a different culture of following SOPs and compliance among those different groups. And then as you scale up and get into the later portions of the product life cycle, the regulatory burden increases significantly. from the standpoint of FDA, EMA, all of the medicines, regulatory agencies, and the process complexity can increase as well. That just kind of gives you a context for the rest of what I'm going to talk about. Something I did want to mention, and someone at the APSA conference gave a talk about this probably about 3 or 4 years ago, and it really stuck with me because I think it's a great model for thinking about organizations in general, is the capability maturity model. And the idea here is organizations go through this sort of stepwise maturing of their processes over time, where initially things are very poorly controlled, very reactive, very unpredictable. And then slowly, as processes get defined and measured and controlled, they mature into hopefully a point where they can engage in process improvement. And in a startup, you're really living in that kind of 1 to 3 zone, anywhere between absolute unmitigated chaos and things are fairly well defined, but still not really to that point of being measurable and fully controllable. So I want to preface this slide by saying this is a vast overgeneralization. So those of you in big pharma, don't get mad at me. But I just want to kind of compare to give you an idea of some of the challenges of the startup environment between academia and large pharma. A little bit unusual in that I'm currently at the University of Florida. So I went from industry to academia. It seems like most of us go the other direction. So it gives me a little bit of a unique perspective. So in terms of processes, procedures, and systems, so that can be your SOPs, your policies, your ways of working. Large pharma and academia, I think in general, tend to have those fairly well established. Again, it depends on the organization, but I think in general they do. Startups, maybe not, right? The startup I work for, we had some basic procedures in place, but a lot of them were untested and they needed a lot of updates over time. Safety culture as well. I think large pharma and academia tend to have a pretty decent safety culture. The issue with a startup is that a lot of times it's not a matter of having a bad safety culture, it just hasn't been established. And then in terms of complexity, for large pharma, typically a given site's going to be working on a couple of projects. Again, this is highly variable, but you'll be pumping out maybe 3 or 4 or 5 commercial products from a manufacturing site. Academia, you have a lot of PIs, a lot of labs, a lot of containment levels, a lot of different types of work, so the complexity tends to be higher. And then in a startup, depending on the startup, you can be working on a wide range of modalities, types of clients, complexity of work. So a startup can end up having kind of the worst of all of these in terms of the conditions under which you're working as a biosafety professional. So that can be definitely a challenge. Some other internal challenges, and these are along the same lines, is you're building the plane while in flight. You're in a situation where you need to develop your processes, your procedures, your systems for how you do risk assessments, how you do training, how you communicate hazards and risk assessments while you're actively working. You don't walk in the door and have all that stuff made for you. If you're lucky, you do, or at least somewhat advanced to the point where you can just make some improvements on it. But that's a challenge. I think the biggest challenge is that biosafety is culturally obscure a lot of times in these smaller companies where you have people that may or may not be from the industry, may or may not be from academia. We had a bunch of people from Amazon Web Services that had joined the company and were in various positions, not necessarily directly related to work in production, but work supporting production. And do you think they know what biosafety is, especially in the context of biomanufacturing? Absolutely not. Then, of course, startups are a pressure cooker, long hours, a lot of high expectations and tight deadlines, sort of like Matt was alluding to. So you get all that taken together and it can be really challenging. And then there's just kind of the general industry challenges. Clients and the client work product, you have everything from tiny, tiny, tiny little startups that want to develop a process to produce phase 1 clinical trial material where, they have very little product knowledge from the standpoint of developing a bioprocess and maybe aren't familiar with the fact that they need to get, or we need to get a USDA interstate transport permit to receive their product, for example. And then you have larger clients, big biopharma clients who have a lot of process knowledge going into the project. And then you have the medicines agency regulators like FDA, and they put a lot of pressure on the organization and really kind of distract sometimes from the safety things that you're worried about. And then everything that comes with the industry, the boom and bust funding, the layoffs, the rehiring, the layoffs again, a lot of churn in personnel, which can make it really difficult to have a consistent translation and transmission of tribal knowledge down to folks in the organization. So what do you do in this type of environment? How do you make good decisions? Internal networking seems kind of obvious, but in a startup, it extends further out than you would expect. So business development is a huge group to communicate with, and one that once I really got in communication with business development and made sure we had a good back and forth, it improved my life as a biosafety officer so much to make sure we weren't accepting projects that would cause a huge biosafety headache down the line, which is another thing that Matt alluded to. And then program management, regulatory affairs, quality assurance, procurement, legal, all these groups, some of which you wouldn't necessarily need to interact with on a daily basis in a larger company. They become really critical in a smaller company. And part of the reason for that is people wear multiple hats. As biosafety officer, I was wearing multiple hats. I was responsible for our export control compliance program. So there's a lot of networking you have to do more extensively than you might in a larger company. And then taking an integrated bioprocess safety approach, I think is really helpful. A lot of the hazards located in a manufacturing suite aren't biosafety hazards, and they can be extreme. Hot steam, really large quantities of hot steam and hot water, working at height, using industrial lift equipment. All of these things are a lot of times equally or more of a hazard than the biohazard itself. So if you take all these together and have an integrated approach to your risk assessment, I think it serves you well because it puts biosafety at the same level as managing a lot of these other hazards that people are more familiar with managing at other organizations. And then using shared tools. So we had a risk assessment process that integrated biosafety risks, environmental health and safety risks, quality, business risks. So we had kind of that same integrated approach with our risk assessment. And that really served us well because it became a good collaboration and communication tool. It also uncovered hazards that interacted with each other, timelines that could potentially have an impact on safety, for example, which are business decisions related to EH&S decisions. And I think it improves the culture too, because it encourages people to think holistically about the way that we approach risk assessment. So I'm going to skip the case study because we're getting towards 2 o'clock so that we can get to Q&A.
Thank you very much, Jason. We don't have questions in Q&A, but we have some raised hands. Okay.
Kendra asked, what is the case study? I'll tell you the case study. The case study was a foreign client who had a viral product which was based on a risk group 3 agent, which was also export controlled. And so we signed on the dotted line before we understood the fact that this was going to be a nightmare from an export compliance standpoint because it's a foreign client. We're going to have to export product to them. So that became a real challenge. And that's around the same time where I really sunk my teeth into our business development folks and said, look, we need to have discussions as early as possible before we bring work in the door. And from there, it worked really well. I would get contacted by our BD folks all the time saying, hey, we're engaging a client about X, Y, and Z. What do you think? And I'd say, well, consider this, consider that. And that worked out pretty well. So.
Thank you. Agrawal, I unmuted you. You had a question. I'm sorry if I'm mispronouncing the name. You can speak. Nope. Okay, it means I probably. We have someone in Q&A. So question, what would be your recommendation as experts on your field for a new biosafety professional? Could you have done anything differently?
Who wants to take that first?
I'll go first. So I kind of cut my teeth in biosafety at a public land grant university, and I'd say that's a great learning environment. Like, get exposed to as much different science as you can. You know, at a land grant, you have animal research, you have engineering, you have chemistry, you have, you know, biology and biomedical type research. I think that broad base has really been helpful for me because you don't get phased when you come up with something new that you haven't seen. I mean, it's you don't have a steep a learning curve. You at least have some knowledge about different areas.
What about you, Brad?
I don't know. I was actually gonna pass that to Katerina. I bet she'd have some good advice. She's definitely had an interesting path.
My only word will be, don't try to fix everything at once.
Yeah.
Do it step by step, slowly, or it will be very tough.
Yeah, and triage according to risk and communicate your triaging to stakeholders and the things that people want fixed, because otherwise it will seem like you're not doing your job.
And I guess my one piece of advice would be, wherever you go, whenever you interview, make sure that you have a good leader there that you can learn from. If you go somewhere where you are kind of thrown in and on your own, or you don't have good people to rely on, you're not going to learn anything. You're going to get the years on your resume, but you're not going to get the actual experience, and you won't be able to take it to the next level until you've actually learned that practical thing. So make sure you have a good mentor wherever you go.
Yeah, and just you know, kind of taking off what you said, Brad. I think, you know, if you are at an institution where you don't have that mentor, I think ABSA, you know, reach out to your network with people and find a mentor someplace else that you can learn from.
Yeah, maybe one more thing. Remember that engineering and facilities staff are your best friends.
Also agree with that 100%.
Yep.
I shared an office with our engineering and facilities manager, and he was my go-to for just about everything. So.
Yeah, I think the one thing that I've learned in kind of just going back to Barbara's question is that build relationships with everybody. You touched on that, Jason, with BD, you know, business development, but the engineers, the facilities people, you know, scientists, you know, you build those relationships and they feel more comfortable come to talk to you, ask you questions.
Yeah, I think silos are bad in any organization, but especially in industry, especially as a safety professional where you have limited resources, limited personnel, you have to build those relationships with people so they know who you are, they know when to call you, and the kind of resource you are for them to, like Matt said, biosafety and EH&S decisions are business decisions, and helping people understand that is really critical to advocating for yourself.
Excellent. Thank you so much. And thank you very much for everyone who attended. I can see one. No, no other. Yeah. Yeah, it's a comment. We probably have to leave. So regarding sharing presentations, we will discuss it with the presenters, but we will definitely share a recording. And we will try to generate certificates. And once we have this event approved by APSA, we will let you know. So you could apply for credential maintenance points. Again, thank you so much for to everyone. If there are no other questions, let me check. Yeah. Thank you.
Thank you all.
Thank you.
Thanks.
Thank you. Bye.
I guess one of us should hit end event.
Yeah, I can. That's fine. I figured Katerina would stay on.
Yeah.